GR IAS Academy.
Hello everyone, welcome to GR IAS Academy. Today we are on the day 5 of Mission Surya Programme. It is a 100-day prelims marathon program in which we will be covering 100 important current affair themes for prelims 2026. Let's see the first topic for today. So first topic is World Ozone Day. Ozone Day is celebrated every year on 16th of September. It is to commemorate the signing of Montreal Protocol in the year 1987. So the Ozone Day celebrations have begun from 1995. So 2025 has marked the 31st Ozone Day. The theme for this Ozone Day was From Science to the Global Action. So this theme emphasizes on the importance of scientific discoveries and scientific evidences in promoting the global action against the ozone depletion. So this was the theme for this year, and in India the celebration was organized by Union Ministry of Environment, Forest and Climate Change. So let's see a few details regarding ozone and ozone depletion. First, let's see the types of ozone. There are two types of ozone. First one is the tropospheric ozone and the second one is stratospheric ozone. We all know that troposphere extends up to the 10 kilometers above the Earth's surface. So some ozone is found here. This is formed due to the emissions released by human activities. This ozone present in the troposphere acts as a greenhouse gas and it is also an air pollutant. So this ozone is the bad ozone. And this tropospheric ozone is also called as ground level ozone. So the ground level or the tropospheric ozone is the bad ozone. Whereas the ozone present in the stratosphere, that is the stratosphere extends from 10 kilometers up to the 50 kilometers above the Earth's surface. Here in the lower layers of stratosphere we have an ozone layer that is say from 15 kilometers to up to 20 to 25 kilometers above the Earth's surface. This ozone layer is naturally occurring one and it protects the Earth from the harmful ultraviolet rays from the sun. So this ozone layer present at the lower layers of the stratosphere is called as good ozone. And we are actually concerned about the depletion of this ozone layer. So let's see the next part. The Champan Cycle. So we have seen that there is a good ozone layer, that is the protective ozone layer present in the lower layers of the stratosphere. Here we have the protective ozone layer. This ozone layer is relatively steady, but within this ozone layer there is continuous formation and destruction occurring. Ozone is continuously produced and destructed within this ozone layer. So this cycle was first discovered by Sidney Chapman and it is named after him, the Champan Cycle. So let's see what happens here. The oxygen present in the atmosphere, when it is struck by the ultraviolet rays, it splits into oxygen atoms. And these oxygen atoms again collides with the oxygen and forms ozone. This is the formation of the ozone in the stratosphere. Whereas the formed ozone, again in the presence of UV, splits into O2 and O. This oxygen atom again reacts with this ozone and produces oxygen. So this way the production and destruction is happening continuously. But this occurs at a proportionate rate. That is, all the ozone produced gets destructed and the destructed one is again produced. So there is always a steady ozone layer present in the stratosphere. Then why are we worried about ozone depletion or ozone thinning if this cycle is happening naturally? That is because of the interference of ozone depleting substances. These ozone depleting substances interfere with the Champan cycle and accelerate the destruction process. So here they act as a catalyst and speeds up the destruction process. So the destruction happens at a rate far beyond the recovering capacity or the recovering rate. So that is why ozone thinning or ozone depletion happens. So the Champan cycle is the naturally occurring one, whereas when the ozone depleting substances interfere with the cycle, the destruction or the depletion happens. Let us see few more details about the ozone depleting substances. So ozone depleting substances are nothing but chemicals that are produced due to various human activities on the earth. They go into the stratosphere and react with the ozone present in the stratosphere. So these are few ozone depleting substances. They are chlorofluorocarbons. They are used in the refrigerants and aerosols. And another one is halogens. They are used in the fire extinguishers. Halogens here are majorly bromine and chlorine. And the next, it is carbon tetrachloride. And the other ozone depleting substance is methyl bromide. It is used as a fumigant or disinfectant, used in fumigants. So these are the major ozone depleting substances. So in order to restrict the ozone depletion, we have many international protocols. So let's see these protocols in detail. So first, Vienna Convention was signed in 1985, was agreed upon in 1985 with an objective to protect the ozone layer in general, and India was a part of this Vienna Convention. So under this Vienna Convention, Montreal Protocol was brought in 1987. So this was brought with a clear objective to reduce the usage of ozone depleting substances. So let us see what was done under the Montreal Protocol. So initially, under the Montreal Protocol, the target was to replace the chlorofluorocarbons. These chlorofluorocarbons are the highly ozone depleting substances. So it was planned to replace these chlorofluorocarbons with hydrochlorofluorocarbons. This is an ozone depleting substance, very harmful to the ozone layer. This hydrochlorofluorocarbon is also an ozone depleting substance, but 90% less harmful than the CFC, so the chlorofluorocarbon. And then it was planned to slowly replace this with hydrofluorocarbons, that is HFC. So this HFC is a not an ozone depleting substance. So this was the planned cycle. Initially, under the Montreal Protocol, we have planned to replace the chlorofluorocarbon with the hydrochlorofluorocarbons. So later, the transition was to replace the hydrochlorocarbon with the hydrofluorocarbon. So here there is no chlorine and it is not an ozone depleting substance. But later we have found that this hydrofluorocarbon, though it is not an ozone depleting, it has got high global warming potential. So this is a strong greenhouse gas. So after this discovery, this Montreal Protocol, an amendment was made to the Montreal Protocol. That is the Kigali Amendment, made in 2016, where we have planned to replace the hydrofluorocarbons with hydrofluoroolefins or other alternatives. So this replacement was planned under the Kigali Amendment. So these are the details with respect to these three conventions, and this Montreal Protocol was the most successful protocol because it is universally ratified by almost all the countries. Why is this the most successful one? Because there were clear-cut timelines in the Montreal Protocol. It was planned to implement in a time-bound manner. And there is the principle of CBDR, that is common but differentiated responsibilities. Not every country is urged to transition directly from the CFC to HFO. Countries were encouraged to transition according to their own financial and technological capabilities. And also a multilateral fund is set up under this protocol in order to provide the financial and technological support to the less developing countries. And another important factor why this protocol is successful, because ozone depletion, unlike climate change, is seen as an immediate threat by various countries, because when ozone gets depleted, harmful UV rays directly enter the earth, whereas the climate change happens in a slow rate. So this is seen as an immediate threat, and countries have acted promptly. This is all about the Vienna Convention, Montreal Protocol and Kigali Amendment. So if we see here, these two were universally ratified and hence they are legally binding within those countries, and this is a general convention. And let's see India's achievements under the Montreal Protocol. India has become the signatory to the Montreal Protocol in 1992. So we have seen the transition planned in Montreal Protocol. First, the plan was to replace chlorofluorocarbons with hydrochlorofluorocarbons, and then with hydrofluorocarbons, and then with HFO, hydrofluoroolefins or others. So if we see the India's achievements, in 1992 India has become the signatory, and by 2010 we have achieved this step. We have completely replaced CFCs with HCFCs. And now currently we are in this step. By 2025 we have replaced 67.5% of HCFCs with HFCs. And under the Kigali agreement, we have to replace the HFCs with HFO, and we have aimed to replace this by 85% by the year 2047. So this is our plan according to the Kigali Amendment, and at present we are in this stage. So these are the India's achievements with respect to Montreal Protocol. So there is a quick comparison between chlorofluorocarbons and hydrofluorocarbons. This was the initial plan under the Montreal, and this was to be replaced under Kigali. There is chlorine here, which is the major ozone depleting substance, and there is no chlorine present in this. This is not an ozone depleting, but whereas we have discovered that it has got high global warming potential, approximately over 14,000 times greater than CO2. So this is a very strong greenhouse gas. That is it. Let's move on to the next topic. So that was all about the ozone and ozone depleting substances. So next topic for today is fungi. Let's see some important characteristics of fungi. Fungi are eukaryotic. Eukaryotic means there is a well-defined membrane to the nucleus. There is a nucleus which has got a membrane. Unlike prokaryotes which do not have any membrane bound nucleus. So fungi are eukaryotic and they are also heterotrophic. Heterotrophic means they depend on others for food. They do not produce food by themselves. They lack chlorophyll and they are non-photosynthetic, so they do not produce food by themselves. And the cell wall of fungi is made of a material called as chitin, whereas the plant cell wall is made of cellulose. Here the fungi cell wall is made of chitin. And the body of fungi consists of hyphae and mycelium. Like if we take a mushroom like this, under the soil there are thread-like substances. These thread-like substances are called as hyphae, and this entire network is called as mycelium. These are the general characteristics of fungi. So based on these characteristics like the heterotrophic nature, absence of chlorophyll and the presence of chitin, we can say that fungi are closer to animals than the plants. So let's see another type of fungi, that is the marine fungi. Fungi not only live under the soil or on earth, they also live in the oceans. In oceans they occupy various environments like from rocky seashores to deep waters. And the marine fungi are actually microscopic, they are not seen by the naked eye. They are very important because they comprise 5% of the total oceanic biomass. So out of the total oceanic biomass, 5% is constituted by the marine fungi, and they play a key role in the decomposition. They play a key role in decomposing the marine organic matter. They form the symbiotic relations with algae and corals. Like they supply food and nutrients to the algae and corals. And they are also helpful in the production of bioactive compounds. So marine fungi have had antibiotic and anti-cancer properties, so making them valuable for the pharma and biotech industries. This is a very important fact. They constitute 5% of the total oceanic biomass. So let's see a few applications of fungi in the biotechnology. Here, let's see the biocontrol. Biocontrol refers to the control of pests or insects using the living organisms like bacteria or fungi. Like the pests or insects are controlled by the living organisms instead of chemicals like pesticides or insecticides. So when we use fungus to control the pests or insects, that is fungal biocontrol. So there is an example here, Myrothecium verrucaria. This is a special type of fungi. An enzyme is extracted from this fungi, and the nano formulation of this enzyme is used to treat the powdery mildew disease in the tomato and grape. So this is a photo, like it shows the powdery mildew disease, and the fungal enzyme is extracted from this fungi is used to treat this disease. And another application of fungi in the biotechnology is that fungi can act as a catalyst or also a feedstock in the biodiesel production. So how are biodiesels produced? When oils or fats reacts with alcohol, like say methanol, in the presence of a catalyst forms biodiesels. So this process is called as transesterification. The oils and fats are converted into biodiesels through an alcohol or a catalyst. And biodiesels formed by this process are called as fatty acid methyl esters, FAME. And this process is the transesterification process. So in this process, as some researchers from Assam have developed a fungal catalyst to speed up this biodiesel production reaction. And we also have a special fungi called as mucorrhinian fungi, which is an oil producing one. This fungi is grown on the agricultural or the food waste, and this fungi actually accumulates the oil, and that is used as a feedstock here in this step in the biodiesel production. So these are some applications of fungi in the biotechnology. Let's see the last topic for today. That is Sattvic NC9. So such term based topics are frequently asked in UPSC. What is Sattvic NC9? It is a chickpea variety. So it is a climate smart, drought tolerant, high yielding chickpea variety. It's basically a bioagricultural advancement aimed at food security and climate resilience. So it is developed to grow in various climatic conditions. It is now approved for the cultivation, and it is suitable for the cultivation in various states. So it is developed by National Institute of Plant Genomic Research, New Delhi. So just remember, Sattvic NC9 is a climate resilient crop. It is an Indian chickpea variety. So that's it for today. For detailed notes regarding these topics, please visit our WhatsApp and Telegram channels, and also subscribe to GR IAS Academy YouTube channel for more updates regarding the Mission Surya program. Thank you.